Okay, I'm going to take a few moments to talk about a lab that you're about to do as part of your molecular genetics unit. It is a bacterial transformation lab. Bacterial transformation is a process that you need to understand. It can happen either naturally, bacteria can go through transformation naturally as part of just one of many processes they do, or we can force them to go through it in a lab situation. PGLO, the other term in this lab, is the name of a gene. This is the name of a gene that codes for a protein that allows organisms to essentially glow in the dark. It's a little more complex than that, but essentially it's a glow-in-the-dark protein. So PGLO is the gene. The protein for which it codes is called GFP, green fluorescent protein. Okay, so this is the protein, this is the gene that codes for it, and in order to make bacteria glow in the dark with this gene, we're going to make them undergo a process called transformation. So, we're going to go over some of the major concepts of the lab in this little podcast, and then we'll go over what you will be doing in the lab itself in the next one. Major concepts you need to understand are, first of all, the significance of bioluminescence or biofluorescence, and this is actually what we're dealing with. In bioluminescence, organisms actually create their own light. They produce light. In this case, biofluorescence, what the organisms are producing is a protein that will give off light if light is shown upon that protein. So it is not lighting up itself, but it is giving off light when, in this case, UV light is shown onto the protein itself. So that's fluorescence. In nature, both of these things are used by real organisms. We see it in like fireflies and whatnot. However, it is also come to be used in the lab. In nature, we don't see it a whole lot on land. Where we see it more often is in the water. Aquatic organisms like this comb jelly or even this real honest-to-goodness fish use bioluminescence frequently. Many organisms that are aquatic can do this, can produce these chemicals that either glow or light up or fluoresce in the dark. We have co-opted this gene to use in different biotech studies or projects. You can see here that these little mice, at least 3 of the ones in the picture and the tail of a 4th, have been genetically altered. They have had one of these genes placed into their own genome and they can glow. There are many uses for this ability. To have a glow-in-the-dark pet, yes, that would be really cool, but what use is that? Well, there are many uses for these proteins, the most valuable of which is to actually be able to see cellular processes happen in real time by attaching this green fluorescent protein to other proteins in cells. We can watch processes, microscopic processes, and see them happen that we couldn't see without this protein. So it's opened up a whole world of understanding for us, and is continuing to do so. And hopefully we'll see a little bit more about that as we go. Let me make sure you understand this concept. We've already spoken of it. PGLO and GFP are 2 abbreviations we'll be using. Please remember your kind of central dogma idea. You have a gene in the DNA. We transcribe that gene into RNA, which then is translated into a protein by the ribosomes, and that gives us a trait. The protein gives us some visible trait. In the case of what we are studying in this lab, our gene is the PGLO gene. It is transcribed by the bacterium once we put it in there. It is transcribed into RNA. The RNA is then translated into the actual protein called green fluorescent protein, or GFP. And the trait that we get when that protein is made is we get glowing of the organism in the presence, when it is in the presence of UV light. Okay? Now, we need to really make sure you understand what a plasmid is, because plasmids are the means by which we can get traits from one organism into a bacterium. Okay, and this is used in many ways, not just with this glow in the dark, but we can get all kinds of genes into bacteria by using plasmids. Okay, understand this drawing. The outline in blue here is a bacterium. This is the bacterium's main chromosome, this big pink mess. If you took it and kind of straightened it out a little bit, you would see it was one great big ring of DNA with all, or almost all, of the bacterium's genes in that ring of DNA. These smaller rings that we see inside the bacterium are plasmids. Okay? Plasmids are small rings of DNA that have just a few genes, a very few genes, and oftentimes what is carried on these plasmids are genes for things like antibiotic resistance. Okay, so when we talk about antibiotic resistance, we need to understand what an antibiotic is. An antibiotic is a chemical that kills bacteria. Antibiotics occur in nature. Fungus are the first organisms in which antibiotics were discovered. Antibiotics kill bacteria. If you are a bacterium, then it would be good to have the ability to produce something that destroyed antibiotics, and they do. Bacteria, certain bacteria, have genes that produce chemicals, proteins, that make the bacteria antibiotic resistant. Okay? So, and very often such genes are contained on these little supplemental, these little accessory DNA molecules called plasmids. Okay? So, let's say that this is a plasmid, and on it, it's got a gene for antibiotic resistance right there. Then this bacteria would be resistant to an antibiotic in its environment, and that would be an advantage. The thing about these plasmids is that they can exist in the bacteria's environment. They can just be laying there. Maybe they were left over from some other bacterium that died. And we can, sometimes the bacteria just take these foreign plasmids into their unicellular bodies, and any genes that were on this plasmid could get used. Maybe it's a gene that this bacteria didn't already have, a resistance gene for a different antibiotic. And so if this bacteria took it up, then it would be a big evolutionary step for it, because now it would be resistant to even more antibiotics. The act of a bacterium taking up foreign DNA, a foreign plasmid, if you will, is called transformation, because from that point, that bacteria would be transformed. It would have new traits based on whatever genes that plasmid carried in with it. So this is the essence of bacterial transformation. Now, as we talk about plasmids, there are many ways to look at plasmids. We can diagram them, as we see here, two different diagrams of the same plasmid. We can actually photograph them. This is a scanning electron micrograph showing the plasmid is curled up. It doesn't look like a circle. In reality, if we straightened out, it would be a circle. What we will find most useful is these kind of cartoon diagrams of a plasmid. This is the P-Glow plasmid that has been engineered for us by a company named Bio-Rad. Okay, and Bio-Rad basically takes the plasmid of the bacteria, okay, and they cut it. They cut it open with restriction enzymes. So let me, sorry. So they cut it open right there with a restriction enzyme, let's say. And then that plasmid could open up like that. And then into that plasmid, Bio-Rad inserts, sorry, I hope I can get quicker at this, will insert a foreign gene, in this case the gene, the PGLO gene that codes for green fluorescent protein. They insert it there, and then they ship this engineered plasmid to us. Okay, they actually ship a bunch of them in a teeny little vial. And so they ship this plasmid to us, and then we are hopefully going to try to make bacterium take up this plasmid. Okay, so just so you understand the diagram a little bit, this is the PGLO gene. It's coding for GFP, green fluorescent protein, and that would be foreign. They would have put that in. It obviously wasn't there from the bacterium. This thing is coming from a jellyfish. Okay, this is a gene for antibiotic resistance. It is actually resistance to ampicillin, which you may actually have taken at some point in your life. Well, this is resistance for that. Okay. And this up here is an on-off switch for an entirely different gene. It's an on-off switch for a gene that allows a bacterium to digest arabinose, which is just a sugar. And we'll talk about why this gene is present later on. This is a more detailed drawing of the plasmid showing you various restriction sites, places where the plasmid can get cut by different restriction enzymes. It's just a more detailed drawing than this one. Okay. Now, bacterial transformation is what we are doing in this lab. What we will do is we are given by Bio-Rad some bacteria, some normal E. coli. Well, they are highly genetically engineered E. coli. They are very weak. They are not disease-causing in any way. They won't survive outside the lab. But this is the bacteria, and they are completely normal. They don't have anything funny about them. Okay? We will expose them to conditions that cause them to more readily pick up plasmids that might be in their environment. But we don't want them to just pick up any plasmids. We want them to pick up the plasmids that Bio-Rad has sent us. They've sent us our PGLO plasmids, and that's being symbolized here by these rings with the little red gene in them. That's the PGLO gene. So what we do is we take our very normal bacteria and we put it into something called transformation solution. It's really something called calcium chloride. Okay, so here we have a test tube full of PGLO plasmids and calcium chloride. Into that we place just normal bacteria. Then what we do is something called heat shock, where we expose the bacteria to cold, then heat, and then cold again. What that does, when in presence of the calcium chloride, it causes little pores to open up in the cell membrane of the bacteria. And into these little pores, the plasmid can be taken in. Okay? So the whole point of the CaCl2 and the heat and the cold is to cause pores to open up so the plasmid can be taken in. If the plasmid is successfully taken in, which it will be in some, not in all, then we say that that bacteria is now transformed. It's got new genes in it. It's transformed. It's going to express traits that it would not have been able to express otherwise. And hopefully we'll see that because we are going to grow these bacteria on a petri dish that's got nutrient for them to survive on, and hopefully we will see them express their traits of glowing in the dark on that dish. Okay, so that is basically the idea of transformation, getting a bacteria to take up foreign DNA. Okay, here you're just seeing another picture, the same picture we saw earlier, but this time we've got this P-Glow plasmid right there. It has 2 genes on it of interest. It's got the one that's going to code for GFP. It's got another gene that's going to code for ampicillin resistance. The protein that gene makes is called beta-lactamase. You don't need to know that. But when the DNA on this plasmid gives directions to make that protein, a protein gets made, and this one actually gets excreted into the environment so that it will destroy any ampicillin that might be in the environment. It just takes it out. It undergoes a chemical reaction and gets rid of that chemical. Whereas this other gene that codes for GFP just produces that green fluorescent protein, and that just stays in the cell. And then if we take our UV light and shine it on the cell, we will see that green fluorescent protein glow green. Okay? All right. This diagram is just telling you about, you remember I mentioned that we put the bacteria in cold and heat and then cold again. That's called heat shock. And so what we would have in here would be our P-Glow plasmid. We would have our bacteria, and we would have our CaCl2, and we'd have it on ice. Then we're going to set the tube in the warm 42 degrees Celsius water, and then we're going to take it out again and put it back on ice, all in an effort to get those little pores to open up in the bacteria so the plasmid will go in. Okay? All right. I think that is most of the main ideas for at least initially understanding this lab. That's the end of this podcast. I'm going to next do one that's just going to walk you through the steps of the lab real quick, and that's it. Thanks for watching.